So we, quickly learned that just looking at some of these complex disorders and looking at a few subjects wasn't going to do it. So that was really when I started to get involved in some of the data sharing types of things and the data acquisition, joint data acquisitions. So we learned fairly quickly after spending lots of money that most individual sites doing our 50 or 60 or 100 subjects is not really going to get us to where we need to be to understand the subtleties of schizophrenia, addiction, autism, developmental disabilities. So we needed methods for acquiring data in a common way. So the BIRN, the Biomedical Informatics Research Network, emerged to try to come up with sort of standards of how to acquire things in multiple sites. Data sharing mechanisms became available to pool data. So again, staying semi-naive, thought, a bunch of sites, all, understanding how to acquire similar data and sharing that through our, the Nitrix and the NIMH data archives and Open Neuro, and that's going to do it. We're going to finally understand, all of these, different disorders. Lo and behold, for lots of reasons, we still don't know how to, define, what's really going on, in most of the disorders, the mental health disorders, that we're concerned with. So the next thing, and again, motivated by, really some lamentations by Tom Insel at the National Institute of Mental Health, where, the scientists were doing a great job. We're doing, the best job possible. We're writing lots and lots of papers. We're finding lots and lots of findings. We're getting lots and lots of grants, but yet the kid with autism, the adolescent, with an addiction issue, or the Uncle Frank with schizophrenia, still had very similar outcomes, to and interventions than they did, the 30 years before. Gobs of money and gobs of tenures, were given to people chasing, spots of genes or spots of activation or spots of bigger or smaller bits of brain. So the revolution in that case was partly okay, well, probably chasing the label of autism, or chasing the label of Alzheimer's disease, or chasing the label of schizophrenia in these very heterogeneous, very syndrome-y types of things may not be the right label to chase, because these are all fairly heterogeneous populations. So sort of two things pivoted in the early 2010s or so. The move to not really to the RDoC, the research domains of classification, that really try to get away from the diagnostic labels and more to the holistic, functional types of labels that are actually quite common across different disorders. And so instead of looking for autism, look for more behavioral types of things that may have a chance of being linked to specific circuitry and specific genetic combinations. So that was one pivot that happened that was important at that point in time. The other pivot was this concern that again, we're seeing lots of things, but none of them are really replicating. Most of the things that we see and report in our literature aren't reported in a way that can either refute or refute or confirm those particular observations. So really, our literature is not really supporting the way we're analyzing the data and reporting it in a complete sense, so that the rest of the scientific process that we need to confirm or deny or see differences in the findings can't really emerge very effectively. So that illustrated lots of reproducibility issues, and so this whole reproducibility topic kind of blossomed out of that. And JB's talk yesterday, you saw a bit of discussion of that kind of stuff. So BJ already sort of foreshadowed what we're part of here. So my talk today really is going to go through a little bit of what is ReproNim and sort of our position in trying to help, in that, lamentation about, we're not really solving, much in mental health. We'll use some ReproNim philosophy to generate a sort of a framework that we try to apply, and then look at some examples, of that framework. So ReproNim is a NIBIB-funded P41, a biomedical technology research center. Its mission is quite simple. It's to promote adoption of a more reproducible neuroimaging research process. To date, there are lots of places you can store data. To date, there are lots of ways you can do analyses, but we just don't use much of our resources to share and to report and to do our research in a re-executable and a reproducible fashion. So we need to make the barriers, to using these resources lower, so that they're, possibly, to be used. The other important concept is this idea of re-executability. So re-executability at an individual publication level, is really what we're thinking of as the cornerstone for moving things forward. Because if each publication that we do in the future is itself re-executable, that then gives you the basis for exploring more properly the generalizability of that observation. Right now, we tend, I write a paper, I'm using some data and some analysis method. You write a paper, you use different data and a different analysis method. Very often, even though we attempt to look at the similar things, we very often find different answers. So what happens? Well, I publish mine, I get my spot of brain as being important. You publish yours, you get your spot of brain as being important. I say, gee, I don't get the same answer as, BJ, and oh, our subjects were different. Oh, my method was different. So that's okay. That's why that's different. So we both publish our things. We're both happy. Now, when you search for, this disorder, you get region A and region B, both as, being important to this. So the problem, therefore, with the literature as it's, being published is we tend not to publish negative things. And we tend not to, look deeply into the many things that are going on in that publication. So we end up proliferating these, potentially both spurious, false positive findings into the literature, filling up our literature with things that we don't really know what, reinforces itself versus which, destructively, interferes with itself. And we're not doing that in a way that we can then look at this. Hey, BJ, what's the difference, between your subjects? What's the difference between our analyses? And systematically, okay, if I used my analysis on your data, do we see something different or not? If I used your data in my analysis, do we get something different or not? We never study that systematically, so that we just fill up these, spurious differences of findings, never understanding, what that difference comes from. And so that's the whole science. It's not necessarily the biological science, but it's the technology science that underlies this, very massive technology that we're using. But unless we understand our technology and how our technology and its interaction with biology affects our findings, we're never going to do much more than a collection of spurious, false positive, laden observations that kind of fill up the literature as a function of various disorders. So the structure of ReproNim, it has 3 technology research and development projects. The first one, is kind of codified around discovery. In order to discover stuff, we define these things broadly. We want to discover data or software or results. That's predicated on the existence of a pool of information to search in, and it's predicated on that pool of information having some sort of standards of how things are described. Unless people are describing things in a similar way, it's very hard for me to discover your information about schizophrenia and my information about schizoaffective disorder if we've not used some way to understand the relationship between what I'm doing and what you're doing. So there's the discovery aspect of pooling data and using standards of metadata and terminology. Then there's the describe aspect. Anything I do, any data I acquire, any analysis I run, needs to encode that information with these appropriately defined terminologies and metadata, so that we can generate information that goes into this pool. So there's a description of the data we acquire, the results we generate, that need to be described appropriately and is influenced by the needs of the discovery problem. Then once I've described something that I need to do, an experimental protocol, an analysis, I need to be able to execute that description. And I need to be able to do that across different platforms. It may be on my own PC here. It may be on my local high performance system. It may be on some national high performance system. I need a way to execute a described analysis, and I need to be able to apply the descriptions that are appropriate to those results, so that they fill this pool that the TRN1, the discovery system needs in order to go forward. And that's kind of illustrated here, that I have some data. Maybe it's my own data that I generated from my scanner. Maybe it's data I fetched from some public place. I have some analysis description. I have some compute environment that executes that. I generate some output. That output needs to be described, and every time I do this, I'm generating metadata and provenance about the process I did and about the results that I generate, and you keep those locally. And traditionally, yeah, we do a good job of, keeping our, BIDS files or our, Nifty files and things like that, but we don't do a good job of holding on to the metadata and the provenance about what we did. And so this is one of the big areas that ReproNim works on, and what we've been talking a lot about is how to generate the metadata and provenance and how to hold on to that locally so that your, provenance and metadata are connected to the data that you have in ways that, connect, all these informations, together. Then you do this, you do some sort of study, you publish, or make available, your data some way. Again, we traditionally think about this in this, lower axis here, where you publish your data to OpenNeuro, or, the NIMH data archive, makes you stuff, your data there for some reason. So that data goes there, but we also lose, again, this metadata and this provenance data. So we're creating, storage, public, storage repositories, that, parallel, this image data with the metadata and provenance data, so that when you go and write your publication, there's ways to connect, not only this publication to the data and where it may live at OpenNeuro or, the NDA, but also all of the metadata, the complete, analysis of the provenance of how each of those elements were generated, of the, the provenance of the statistics that were analyzed, et cetera, in a way that are machine readable and also humanly accessible, so that that can be accessed, reused in other situations, confirmed, and then extended or studied, systematically, as I sort of foreshadowed before. So that's kind of, where we want to get. So in our first 5 years of thinking about this, we've generated some philosophy. We really want people to try hard to plan everything ahead of time. Very often, we jump right into a study, and collect all sorts of things, and then later think more about what it is we want to do, and find, maybe we didn't, acquire everything we needed, and then you have to figure out a way around, a workaround, for that. So we need, to plan everything, because interoperability creeps in when we do unplanned and improperly prepared for, procedures in our science life. We should be version controlling everything. And that's because irreproducibility creeps in when we can't quite remember exactly what we did, when we did it, and why we did it. And was that last thing I did for the last 10 subjects really the same thing I did in those first 10 subjects? Or did I change something? We don't have the connection. I don't have the, that script, or that version of that software. Did that change? Did my operating system change? Blah, blah, all sorts of stuff happens. So version controlling everything. And by everything, I mean your data. Our software folks are all, used to version control in many ways, but this is more than software. It's your scripts, it's your data, it's your results, it's your processing, everything evolves, and everything evolves for a reason. And we just need to capture and hold on to that so that you can get back to what you did and be sure of what you did, and share what you did. Ultimately, we want to be using standards for everything, because irreproducibility creeps in when it's hard and it's required for me to describe my process of my data, of my analysis, to everyone else. If I just make up my own, data format and, willy-nilly, write my own data down, I'm going to have to share it with someone. And if I just do it willy-nilly, I then have to take the time to explain to BJ, hey, I put, the ages over in this subfile and the statistical tests, over here, and I have to explain that to you, and you then have to decode that and use it, and that's just very inefficient. If I can use a standard where where age lives, and where the statistical test lives, and where the raw data lives, and what a T1 data is, then it's easy for you to share it, and you can wash your hands of it because it's self-describing because of those standard types of things. So again, it's more efficient for you, and that's where the irreproducibility creeps in when you don't want to do it, and it's too hard to do if you haven't used standards. Annotation for everything. So it's all, irreproducibility creeps in when we can't understand what we did and integrate it with what others have done. So when we make a measurement of something, an activation, a volume, we need, and of a region, or ascribe an activation to a region, we need to annotate these things in ways. We need to annotate our subject populations so that when I try to connect my data to your data, we know what a volume is, and what a activation region is, and what my subject population is. So everything should be annotated so that we can integrate our information better. And everything should be archived. And again, anytime I complain about what the community is doing, my one finger pointing at the community also has the four fingers, pointing back at me. I'm old enough that I've done everything wrong already, so I can't go back to a publication from 5 years ago and be sure that I could find all the raw data and be sure that I could find the operating system and the script that I ran. So again, proper archiving and proper connecting of archiving to the results and to the process that is associated with the paper is important, because irreproducibility creeps in when no one, not even yourself, can find the data and the processing that you used in a particular publication. So ultimately, these are all important topics because everything matters. Exactly what data you use matters. Exactly what software you use to measure things matters. Exactly what statistical model you use matter. Exactly what thresholds you use and what story you choose to tell depends on the types of information that are affected by myriad decisions along the way. So unless we write down or document those myriad decisions, it's going to be impossible to get back to what you did and how that differs from what I did. So we know imaging is complex. There is lots of steps. There's lots of stages to an analysis. These include design of your study, collection of your imaging and behavioral and clinical data, processing of that, statistical analysis and inference from that, and then that publication. So there's lots of stages. There is huge data reduction. Think of a study using ABCD data, the adolescent brain and cognitive development data set, 10,000 10-year-olds being scanned for the next 10 years. There's hundreds and hundreds of terabytes that is at the core of that. You do some analysis and you turn it down, do something, and it ends up being a couple bits of data. This region is different than that region. 1 bit on off for a particular region. So you've done a huge data reduction to get to that conclusion about the few different regions that you're going to claim are different or correlated or informative. So huge data reduction. We need to understand that path that got you to that data reduction precisely, because there was a million different ways to have done that. And if we do a different one of those million different ways, we might get a different answer, and we need to understand that. So that sort of sets up the two axes of this ReproNim framework. We have these stages of a study, and we have these principles that I talked about. So that gives us this sort of framework that we want to be thinking about and keep in our minds. So the Repronum Pathway essentially is a way to navigate through these types of principles and practical study stages. Doing all of these things is hard. As we were mentioning, there's so many, because we have to keep track of everything, there's so many things you have to keep track of in order to really do a process in a way that someone else can sort of truly recreate and benefit from. So as I mentioned, this ReproNim platform, this ReproNim pathway is a way to sort of virtually, affect and travel through the space of your experimental stages and the types of practices that improve our reproducibility. Adding these repro philosophies to an ongoing study, is hard, and it's kind of like
some people like to climb mountains.
A plane while it's flying.
I like to build planes in the air. I grew up wanting to be on a wing, wanting to be up this high. Sometimes
whole video, but you can share that. But if you have a study, you've already acquired some stuff, you've already done something, and I'm sitting here and saying, oh, you should do all these things. Well, you can't. The plane's flying. You can't do your study registration. You can't change the container since you've already done a bunch of things. All these things. So all of these objectives are really modular. So we don't have to do everything. The more we can do some of these things, the better off we are. And if we can pick off easier ones, if we pick off elements of this that can be done later or earlier in the process, our goal is to break up this problem into bite-sized enough pieces that maybe there is still something you can do on this ongoing study. The next study you plan, well, maybe you can't do all of this, but pick a couple of these things and try to work with these concepts, going forward in a more sort of piecemeal way, because ultimately it is too hard to jump unless you're building a center from scratch into all of these things, right from the get-go. So I don't want to dwell too long on the exact tools, but in the study design, one of the key tools is pre-registration and trying to get our studies planned ahead of time and pre-registered, so that sets out a particular scope of an analysis, helps control for false positives, and gives us the basis for publishing negative results, which are very important. And there's tools that help that. It's also important to remember that the ReproNim framework, yes, ReproNim develops some of these tools, but to some extent, I don't really care exactly how you accomplish, planning, a ReproNim tool or not, as long as you're doing something, about planning or something about version control. There's things we have developed to try to help, but again, to some extent, the point is to accomplish these functions one way or another. In the data collection side of things, we talked a good bit, yesterday about data lad, which helps, convert incoming data into standards. And there's, lovely standards that we have, available, to date, BIDS and NIDM and things like that. And there's ways to archive data. There are ways to run, containerized, processing as a standard, so that it's easier for me to hand to someone else the exact workflow I did and not be so dependent on exactly what my local, hardware is. There are ways to, run version control with analyses. There's formats and archives, that relate to that. Statistical analyses, have representations, have standards, have annotations, and archives, and then the results, have places that locally, can be published. The data can be published. The results can be published. Both the metadata and the results. There's a handful of tools there that we've created that sort of help you navigate through this technology. So similar concept here, just sort of illustrating these different stages, and again a number of the different tools that ReproNim adds to the plethora of tools that help you solve these various things. Some of the NIDM and the Repro Lake types of things will be part of our discussion this afternoon, and the containers and data lad were sort of part of yesterday's presentations. Okay, we got us some religion and we got us some tools. Now what? So these tools and philosophies are just ideas, and they're rather amorphous until you see how these concepts can be applied to the real world. So one of the things we're working on is developing a series of documents designed to work through real world examples to document these types of things, and that's sort of why it was important to us to have some hands-on portions of the tutorials that we do, is just to not only talk about philosophy, but to give some real world examples of how some of these things work. Each volume of this series of documents tries to address a bite-sized chunk of analysis and provide a worked example that hopefully you can then evolve that example to work in your particular types of situations. When each of these modules are fully fleshed out, it should be able to define the problem that this little document discusses. It should explain sort of the theory of a ReproNim solution and provide an example in practice of the solution as an example. The most important part of these documents, though, is what did this cost me? What are we asking people to do over and above what they otherwise would do? What did this cost me? What do I have to do that is over and above my status quo? And that's important to acknowledge. But then we also have to be able to assess what have we gained by doing this. And it's really that cost-benefit ratio that's important. That yes, you may have to learn some new things. You may have to slow down for a little bit and change some infrastructure. But hopefully, ultimately, the argument has to be that we've gained something that is important relative to that cost. And if that gain doesn't outweigh that cost, well, then people aren't going to be doing this. So the document, this How Would series, is located at the aptly named how-would.repronim.org. And there's a handful of these volumes that are there now. all marked up in this sort of nice little markup system. And again, can't go through each of these. Some of them have containers and workflows and binder ways to help people go through those in reality, and others have sets of instructions and things like that you can do at home, given that you have the right software. But so again, the high-level summary of the how would ReproNim do local container analysis was very similar to what the containers discussions yesterday were going over. So if you have a container and you already know how to run Docker, great. You tend to use this particular command. In the ReproNim way, you run essentially that same command. So we've not really mucked with that command too much. We just do it in a way that runs that container under provenance and version control. The things we do add is trying to make sure that your data is under version control and that your container is under version control. So there's two little concepts we add to what you'd otherwise do. But in the end, running a container your way and running a container the ReproNim way is very similar. And so again, it's not costing you much, except making sure you have DataLad installed and making sure you commit to version control your input data and your container. But what you gain from doing that is every resultant file you generate now has its history. From that history, every resultant file that you generate can be re-executed by anyone who has access to that data and access to that container. And you gain ease of shareability because you can use, publish, you can copy, that whole data and history chunk, to a place, give it to someone, and they will be able to unpack that, check its version control, and use its provenance in order to regenerate that particular result. So again, without you doing anything other than making the ability to share that result with them. So again, you gain a bunch. Yes, it cost you a couple tools to add and two concepts, but you gain all sorts of things in that. So this is the kind of motivation that we're trying to set up on a lot of the different topics in these How Would series. Similarly, the publishing a set of Nifty files. Well, today, if you had some Nifty files and you wanted to publish it, well, you'd figure out how to get them to Open Neuro or to Nitric or to NDA or somewhere, and it's kind of a ad hoc solution to how you would go about that and figure out how to share a bunch of Nifty, publish a bunch of Nifty files. In the ReproNim sort of DataLad way, again, it's as simple, quote unquote, as getting your data under DataLad control, and getting once it's under DataLad control, DataLad already knows about lots of our sharing infrastructure. Open Neuro, it knows about the Open Science Foundation, it knows about the GIN storage network, it knows about Nitric and things like that. So ultimately, publishing is just taking a copy of that DataLad content and publishing it to some repository, Figshare, etc. So again, that becomes very simple. If you have Nifty data, well, Nifty's good, that's a standard, but you probably also have some behavioral data. So the ReproNim way would say, don't just share a random CSV, and even Nifty, you certainly want to represent it in bids so that the structure of what that data means is clearer to end users. The one problem with bids is that's not very specific about the behavioral and other metadata that's associated with it. So the next level of standardization of that content that you're trying to publish is to add an IDM representation of that metadata. So again, so there's a handful of things to do to add the semantics, to add the standard representation, move that data into a bids format, move that data into a repro bids format that has the semantics, and then use the simple DataLad publish functionalities. So again, the cost again is learning something about DataLad and installing DataLad, and taking the time to commit to labeling, to annotating your behavioral or clinical data. But then what you gain is a ultra ease of shareability, publication of that, and a complete description, that makes that thing that you've shared, completely machine readable, completely other human readable without any intervention, by you. Third, just a quick example is managing local free surfer results. So lots of people have done lots of analysis already and you haven't done it yet, in the ReproNim-ish way. But again, the tools that will be a good part of this afternoon's discussion. Now that if you've heard the earlier lecture, you know more a little bit about the semantic markup and the linked data concepts. Well, applying that to data that you already have. If that data is generated by ants, or if that data is generated by FSL, or if that data is generated by FreeSurfer, there are ways to apply that annotation to those data sets. And so, once you've applied that annotation to your data sets, you can aggregate those across your different studies and do all sorts of fun study things at home. But also, there's lots of publicly available data already, that uses FreeSurfer, that uses FSL, that uses ants. So you then gain ability to integrate your local data with publicly available data. And so, one, you can share your data more easily, and two, you can use properly annotated data from public data sources more easily. So making it relatively trivial to integrate my FreeSurfer data with the Abide or the ADNI or the Healthy Brain Network data, and ultimately other data sources. So again, there's a cost to doing that in terms of the running a tool or two and installing some infrastructure. But again, the gain, the reward is hopefully big enough to not have to solve if 1 or 0 is male or female, and if ADHD dash H is ADHD with hyperactivity or with hypertension, over and over and over again. So that's sort of the high level, delving into a couple of those. And there's there's a beautifully written containerization of a workflow. So again, right now we can talk a lot about existing containers, but most of us need to consider how to containerize our own workflows, since we probably don't do exactly what other people have done. So it's a lovely little example walking through the Enigma shape analysis protocol and turning that into a container. So again, a number of examples. Some are still in progress. Some are still so use them and contribute to them if you find errors. So the last topic I want to get into in the how would ReproNim is really this evolution of what a publication really means. And so I foreshadowed in the beginning that ReproNim's view of the world is really, we want to get all of our publications to be re-executable publications, i.e. repro pubs. So conceptually, what that means is, we need our usual words, because humans do have to read this and humans do have to understand what it is my study was about and what I found. So fine, we're not getting rid of, lovely prose and good writing, but we want to make sure that those lovely prose and good writing is connected to something that is machine readably and specifically connectable to the data that went in, to that, publication, to the workflow that was used, to the execution environment, and to that complete set of results, so that those complete machine readable documents are there to support lovely words and stuff that the human writes about, that the human reads. We also want the machine to be able to follow along and to be able to regenerate those particular results. In part because we tend to write a story about some subset, some storyable subset of our results, and not necessarily all of it. And we don't talk about all the things that didn't show up, and yet the things that don't show up and the parts of the story that didn't fit in and didn't get reported in your story are still important in terms of how your results relate to the rest of the field. So ultimately, a completely re-executable publication requires the complete description, the complete provenance and metadata of the data, the workflow, the execution environment, and the results. So how do you do this? Well, this doesn't really change, the publication per se, but it, can, take advantage of, supplementary data and just being clear about how you, annotate in your paper, all sorts of these important, types of features. So a very simple example of this, just using pointers in the data and supplementary information in a publication, is this simple workflow example. Just the FreeSurfer volumetric analysis across some small number of subjects, but where the data is explicitly annotated and collected from public data into a specific little data set. and that's accessible to anyone via DOI. The workflow specification is a particular NiPipe workflow that lives at a GitHub repo and is accessible by anyone. The execution environment comes from a particular Docker container that's accessible by anyone, and the complete results of that analysis are also shared back at the GitHub workflow site for this. So anyone can get that data, can get that workflow, can run that analysis and confirm that they get the same results. Now it turns out when you do that with the container that we provide, you do get the same results. But if you use your own environment and things like that, you can document that the results are different. So again, this is more the plumbing example of something very simple that you could re-execute if you wanted to. The next stage of this is to doing something more interesting, more complex. So phase 2 of this exercise is to look at something like the structural correlates of IQ in typically developing children. There's lots of papers that have been written about topics of that, so it's very easy to come up with a handful of published observations that give us some starting hypotheses. And using those hypotheses, can we re-explore the known potential methodological dependencies and see if we can confirm or the claims of those particular publications in a now re-executable way. Had all the publications with hypotheses that we were looking at been done in a reproducible way in the first place, well, that would be easy to switch the data to new data and do these kinds of things. But most, almost all prior publications, just sort of say, oh, I used Civet, or oh, I used FreeSurfer, and not too much about the exact, analysis. So anyway, so in this case, questions about IQ in typically developing children, we have to do some data discovery where some relevant structural data that have, IQ and age and sex, do a pre-registration of our specific hypotheses, create the annotation of the data sources if it hasn't been annotated already, particularly the metadata that we need, the IQ, age, sex, etc., from our different sources, containerize a workflow, and execute that workflow and apply proper annotation to the results, and then extract and do the statistical modeling from that pile of results. So this slide here, not to go through the details of it, is sort of 9 different hypotheses that we generated from 3 different publications in the literature. Some looking at sort of brain volume should positively correlate with IQ. Left striatum should positively correlate with IQ in male and female children who are older than 20. Blah blah blah. So a handful of striatal hypotheses, corpus callosum hypotheses, and total brain hypotheses. Just the selection of some random observations from the literature that we could attempt to reconfirm or deny these and do it in a completely re-executable way. So we've done this. Those hypotheses and the data sources that we had were pre-registered at the Open Science Foundation. Since our analyses targeted things like striatal volumes and total brain volumes and corpus callosum volumes, we knew that FreeSurfer and FSL and ANTS could do such things. And again, from prior knowledge, we know that answers can depend on different tools. So in this case, we're trying a handful of different tools so that we can compare and contrast that as well. So there's a NeuroDocker, a container that can be pointed to, and anyone can fetch and get. The data sets came from publicly available data that had age and sex and IQ available, in this case from the Abide 1 and ADHD 200, although since we've done this, there is more data that would be available for this. And the statistical analyses and results, workflows are saved in a GitHub location. So again, anyone can access this data, anyone can access this container, anyone can access our registered hypotheses and research plan, and anyone can access that statistical approach. So just one of the examples, that total brain volume will positively correlate with IQ in both sexes across the complete age range. Well, this example we confirmed with a very similar statistics to the original observation. Not surprising, it's a kind of pervasive finding about IQ and brain volume. It's not a huge correlation, but it's a significant correlation. And in this case, we're looking at due to 479 subjects, that came from the ABIDE and ADHD 200 data set, and fine. So that is an example of a confirmation of one of the hypotheses. This table here sort of shows that overall summary of the different hypotheses, and green for things we confirmed and orangish for things we didn't confirm. So again, this total brain volume was confirmed in all three different tools that we looked at. There's some striatum observations in both genders and in just males that we expect correlation, whereas they didn't see a correlation in the females where we do, so we didn't confirm that because we end up finding a correlation in the females. And similar, we find and not find some of the corpus callosum values. So again, I don't want to really get into the nitty-gritty details of this. The point is, taking a set of hypotheses, we can explicitly test those on different data sets, potentially different tools, and come up with a assessment of did we confirm or not those particular findings. The benefit, though, of doing it in sort of in the re-executable way is that, fine, right now, if I look at one of these disk confirmations, gee, free surfer in these younger than 20 females, the original paper said it wasn't significant, and we think it is. Well, the original one may have used civet and didn't make that available, so we can't really go back and do that. But someone who looked at our finding and wanted to do it again and see if they do or don't find the finding we find, could get exactly back to our observation here and build upon that. And again, so more observations or using similar data would be possible from the framework that we're starting with here. So again, just some pictures of some of the things that did confirm. We expected significant correlations and observed significant correlations in these striatal volume observations. In these corpus callosum, we were expecting a negative correlation and observed not significant correlations in some of these different age and gender groups. So again, just poking around at those particular observations. But again, the nice part about this is it's done in a re-executable way so that one can then explore this relationship as it has to do with tool, as it has to do with subject populations. And again, in this case, we're using different data subjects and different analysis tools. So that's again, part of the reason why it's different, but it's very hard to discern that unless the original publication was also done in a re-executable fashion. So to end sort of this set of observations is we really want to get our observations into sort of a completely linked everything concept. So again, traditionally, you make an observation, you publish the statistic model that you use, you will report in your publication the correlation coefficients and the p-values and things like that. And so fine, you can read that result. In the ReproNim way, in the re-executable way, you can generate a very similar plot, again in this case looking at corpus callosum volume and its relationship to brain volume. But in the re-executable framework, each one of these points knows all sorts of things about it. That point knows who the subject was that generated that particular value. What are the clinical variables? What are the demographics about that? It knows what MRI scan was generating it. And it knows where that MRI scan lives, so anyone can get access to it. It knows what workflow exactly generated it, and where to find that, and what hardware and software that was run on. It knows where the concept of a corpus callosum volume lives in the hierarchy of and relationships of various measurements that one might make, volume as opposed to area, or corpus callosum as opposed to genuine corpus callosum, and things like that. So those relationships can be kept. So in this case, that workflow that generated this was a set of FSL and FreeSurfer and ANTS pipelines that, again, from that point, you can get to the data, you can get to the workflow, you can confirm and regenerate each of the different points that went into that. So by doing that, any person who has regenerate to that container can regenerate that particular observation. So by being able to certifiably regenerate that, you can then explore the generalizability of that by changing the data, by changing the processing, and see what that does to that particular type of observation. That point then is fair in that it's findable, it's accessible, in the Repro Lake, it's interoperable, and it can be regenerated and reused in other operations. And similarly, the statistical analysis is also can be regenerated with the same data, can be compared to identical execution in different data, is version controlled, generate provenance, and accessible to all. So that's kind of the high level introduction to the different types of things that ReproNim is trying to fill in and make more accessible to the greater community. And we appreciate BJ and the whole Yale team of letting us try to discuss these and show some of these tools with you, and hope to be helpful in your adoption of these types of things. I just want to end now with the Derm and Strang of okay, fine, really do I want to do reproducibility? I'll just do it or else. You won't get funding in the near future. There's new policies coming out that really is trying to up the data management process. So again, being able to adopt more and more of these definably re-executable and reproducible processes is going to be beneficial both to your funding and your granting applications. Ultimately, more and more teeth is getting into the annual funding, that if you haven't acquired the data, if you haven't shared the data, you're required to. Again, more impact on your future award of even existing grants is becoming more and more dependent on that, sort of in an analogous way that the publications and getting things into PubMed Central is now quite teethy in terms of getting subsequent money. You won't get published. Again, more and more journals are beginning to enforce more and more their software and data sharing policies. And again, unless you're prepared to be able to handle that efficiently, you're going to end up paying a price for that, and that's amping up over the days and years. And without funding and publications, you're not going to get tenure. Just joking, no one gets tenure anymore. So, and the most important thing is John Oliver and the Late Show will laugh at you if you don't do your process in a reproducible way. I'm not going to play this video clip, but I recommend watching this video clip, and it really laments and is really quite disturbing, the general irreproducibility of science, and quite funny, but in a very sad and scary kind of way. So again, all of this is helped by reproducibility by design. As we go forward and keeping these concepts in mind. ReproNim is a huge group of folks, many of which you've seen in the different presentations, many of which you haven't seen, so I want to make sure they get listed here. And again, it coordinates across a number of existing initiatives as well, and again, a lot of help from the INCF to coordinate nationally and internationally these efforts. And you've probably seen this elsewhere, but we are accessible and need and want to be helping everyone do these kinds of approaches. There's webinars, there's training materials, there's YouTubes that you'll learn more about, because these talks from the Yale series will be there as well. And contact us, we're happy to help. So I think that's where I will stop the official presentation.